students. All right, happy lab day to you. And today we are going to explore various cells under magnification to see if you can identify cells undergoing the various phases of cell division. So to begin, real quick, raise your hand if you can share with us the memory aid to help you memorize the correct order of cell division. I picked my apple tree clean. Hopefully you remember that, and you can compare the first letter of each word with the first letter in each of the phases of cell division. Remember, study all of that for tomorrow's quiz. All right, let's move on to today's lab activity, the cell division lab. And this picture here of an actual slide may overwhelm you at first. But what we're going to try to do, and I'll show you here in a moment, what we're going to try to do is to zoom in on some of these cells. And by the way, any idea what kind of cells these might be? What would make you say that? Plant cells. You can see the cell walls very, very clearly here. So it would be nice if I push the right button. Now, it would be nice if we could zoom in this close and see some really great examples of cell division going on here. In fact, any idea what phase of cell division this might be? What evidence or clue would you use to determine that? Hopefully you're talking about the appearance of these chromosomes moving away from one another, and they move away from one another during anaphase. Well, I'm going to be quizzing you here in a little while with our lab activity, but before we do, I'd like to take you over to our virtual lab here, because I have something set up for us all. Since we're not sharing our microscopes right now, I'm going to share my microscope with everybody. And first, take notice of what I've done to my microscope. I have attached a digital camera in place of what's normally here. And what's normally at the top, everybody? The eyepiece lens. So instead of the eyepiece lens, I removed it and I kind of taped on a digital camera here on the top of our microscope. I've also placed a slide here, which is a number of plant cells undergoing mitosis. And raise your hand if you can tell me what is mitosis. Nuclear division. Very good. So there is a specimen on this slide, which we're going to look at together here in a moment, that the cells are from a plant. In fact, it's from a bean root plant, and it's from the root of a plant because that's where a lot of cells are dividing very, very quickly. And so we have caught in the act a number of root cells undergoing cellular division. So, and in particular, mitosis. And so we're going to check out this slide together. So I'm going to go to our next view here, which shows me in a tiny picture surrounded by what we are looking at under the microscope right now. Do you see how it's moving? That's because I've made the background our view through the microscope, which currently, I'll turn it this way so you can see it a little bit better. Our low objective power is pointing down, so this is under low power magnification, looking at our bean root slide. Now again, It may be overwhelming to look around, but what you will want to do is moving the stage. You want to hunt around. You want to explore. Once you have it in focus, which I already do, you want to hunt around to a place where you see lots of different cells undergoing cellular division. Now you'll notice all the green lines are the cell walls, and the darker red or purplish colored areas are the nuclei. And you can see as we hunt around, and part of it is just exploring your field of view here to try to see some cells doing stuff. And the more nuclei we see, like as we get closer to the actual tip, and I'll go all the way down here, you can see that's the actual tip of the bean root. But towards this region here, there is a big cluster of all of these nuclei. They are busily doing something. So this might be a good place for us to stop and zoom in to the medium power, which is what I'm going to do right now. So I'm going to adjust our microscope, allow the digital camera to compensate for the light. And oh my, I don't see anything yet. It's still blurry. So at this point, we can use the coarse adjustment knob still to pull it a body tube away from the stage to get it into focus a little bit more. We can use our fine adjustment knob to fine tune our view. And wow, check it out. We are actually now able to see a little bit more going on here in our slide. And again, we want to hunt around, move the slide around as we have zoomed in a little bit more to see if we can find, and by the way, this, I don't know where, if we can see it on this image, there's a little spot here. I can't use a pointer at this point in time. Oh wait, here we go. I can use my finger. See that dark area right there at the end of my fingertip? That's just a smudge on the lens. I wasn't able to get it off. Maybe next time I'll get it off. So you can know that because as you move the slide around, that little smudge stays put. So that little smudge is just, it's not part of the slide itself. It's a little speck of dust on the microscope. So we can be hunting around, looking for clusters of cells undergoing something. And we're going to zoom in a little while. Maybe this one here by the smudge, I'll put it right over my head. And now I'll zoom in to the high power. So we'll zoom in to the high power, and I may have bumped the slide a little bit, but that's okay. And it is completely dark, which means we're under high power. We have to go to high light. High power, high light. So I've turned the lamp up a little bit more. Camera just is compensating. Use only the fine adjustment knob to get our view into focus here. And we're getting closer. Getting close. Oh, check it out. Now we're under high power magnification. And we are viewing individual cells at this time. And I'm moving the microscope around slowly, the stage of the microscope around slowly to see if we can pick out some. Oh, look at that. You can see some chromosomes lining up there. Think about what phase that might be. Part of this activity involves spending time hunting around, simply looking for appropriate slides, or rather specimens under magnification in order to find cells at different stages of cellular division or nuclear division that we call mitosis. Here's another one by the smudge here. You know, that smudge makes a pretty good pointer for our purposes here. But notice, you know, these squiggly lines, which are the chromosomes, we can tell that they are doing something different here than, say, in a cell nearby, like here or here, where the smudge is. So rather than me taking all the time to hunt for specific cells, I've already done it before, and I've taken some pictures. And so I'm going to head back to some pictures here. I'm going to go back to our PowerPoint here, and I have picked out four different cells that I have drawn arrows next to. And it's these four cells that we are going to use for our lab activity right now. And so what I'd like you to do is to take some time in your lab page book, or the page in your book for the lab, and I would like you to illustrate only these four cells. This cell, this cell, this cell, and finally this cell. The four cells that are being pointed at or pointed to with the white arrows. So you can pause me now and take several minutes to illustrate or draw just those four cells, not the cells surrounding them. And while you're drawing them, think about the diagrams that you drew from yesterday, and think about what these chromosomes are doing during these four stages of cellular division. So pause me now. All right, hopefully you've had plenty of time to illustrate these four different cells in your book and have made an attempt at identifying what phase of cell division you think was going on. So I am going to point to each of these cells, and I want you to yell out your answer, and then we'll review and see how well you did. So let's start with the first cell example. Just yell it out. What do you think is happening during this phase? What about this phase? Next, this phase. And finally, this phase. Well, let's see how you did, because I examined them very closely when I took the pictures and put them together and examined them under perhaps better clarity and magnification than what you can see on the board. So let's see how you made out. Here we go. The first example, this is actually prophase, and the chromosomes inside are starting to pair up. Prophase pairing. Here we have pairing of chromosomes taking place. In the second example, notice the chromosomes are meeting in the middle. Metaphase is when they meet in the middle. Metaphase. Third, we have them going away from one another or moving apart during anaphase. This is anaphase, moving away. And finally, during the fourth one here, they are still surrounded by the same cell wall. However, we have two new nuclei being formed. So two new nuclei take place during telophase. So how did you do? How did you do? How many? Raise your hand if you got all four correct. Awesome. How many of you got three correct? Two correct, one correct. Well, if you got all four correct, you also got one correct. Even if you only got one of them correct, perhaps the most obvious to you, I will consider this a success, the lab activity today. But before I bid you adieu, make sure that you finish your conclusion and then use the remainder of your time to study for tomorrow's quiz. So for now, I'll say bye-bye.